Hey all, it's Dr. Rivera, and now I want to go over meiosis with you. So we're going to start out with the same cell that we started out with in mitosis. We've got 3 homologous sets of chromosomes, 1 set from mom and 1 set from dad. Mom is in hot pink, dad is in green. So each parent is going to contribute a haploid set of chromosomes to make a diploid organism. So this is where we start out with the whole thing. Now, the first step in meiosis is not going to happen until after DNA synthesis. So before meiosis even starts, we have S phase. So let's double everything right now. Okay, so we're going to have, I'm just going to draw these as X's. It's going to be 2 copies of number 1, 2 copies of number 2, and 2 copies of number 3. Now, just a reminder that one of these X's is still called a chromosome because of historical reasons, but it's made up of 2 chromatids. So this here is equivalent to this chromatid right here. It's the exact same thing. Now, keep in mind last time what I told you, that these guys are going to be connected. They're connected along their length, and they're especially connected right at their centromeres. I like to draw them connected right at the centromeres. So we're also going to have a replication of each of the chromosomes contributed by mom, 1, 2, and 3. So now during meiosis, the cell is going to need to accomplish 2 really big things. So one is to halve the number of chromosomes. So we want half as many chromosomes as in the starting cell. So we're going to go from 1 cell to 4 cells. In mitosis, we go from 1 cell to 2 cells, right? We replicate the DNA, divide it up in half. In meiosis, though, we're going to replicate the DNA and then quarter it. So we're going to make 4 cells out of this, which is going to halve the total number of DNA. We're going to get back a sperm or egg contribution, basically. So what's going to happen here? Well, the first trick to understanding how meiosis works is to realize that this connection here that is holding together the 2 sister chromatids, the identical copies of dad's number 3, those don't get broken until the very end. So we've got 2 rounds of cell division in meiosis, and the first round does not separate these sister chromatids, okay? So the first division in meiosis is going to divide up the homologs. Now what's really cool is that the homologs can divide up in a few different ways. So you could get all of mom's on one side and all of dad's on the other, or you can get more mix and matchy. The cell doesn't care. So we could end up with a different situation where we've got mix and matchy. We've got a mom's 1, 2, and 3. We could also have, and we're also going to have a dad's 1, 2, and 3. Okay, so any combination you can come up with, the cell can do. What's the rule here? The rule is that each side is going to need to have 1 chromosome of each type, a 1, a 2, and a 3. So 1 homologous set of chromosomes on each side of this metaphase plate. So during meiosis, we're going to have lining up of homologs. So in both of these pictures, I'm trying to represent the homologs lining up. Homologs line up. Now the reason why the cell is going to do this is to make sure that each of the daughters, they're getting a complete set of chromosomes. If these homologs do not line up, then we can have really bad situations happening, and I'm going to show you guys what could happen. Then I'm going to draw a big X through it because this is not what happens. If we just put 3 on each side, but they're a random 3, right? So we don't have homologs line up. We just make sure we get the right amount of DNA. Okay, so let's just have some of mom's and some of dad's on each side. Let's say that this is dad's 1 and dad's 2 and dad's 3. Okay, so this is going to be not homologs lining up here. We could have moms 3. Whoops, that one didn't work. Moms 3, moms 1, and moms 2. So you can see what's going to happen here is you are going to end up with 2 cells that have the wrong sets of chromosomes. You're going to have one cell that has 2 copies of 3 and one of 1 and none of 2. Over here, this cell has 2 copies of chromosome 2, a copy of 1, and none of 3. Neither of these cells would be functional, right? So neither of them is going to have the right numbers of chromosomes of each type, so the right genes to run a cell. They're going to be missing stuff. The cells that this one produces after the next round of division will not have any of the genes that live on chromosome 3. This one won't have any of the genes that live on chromosome 2, and they will not be able to make a functional zygote when they undergo fertilization. Okay, so this doesn't work. No. And the big problem here is that we do not have homologous chromosomes lining up. We've got 1 lining up with 2, 3 lining up with 2, 3 lining up with 1. And so this kind of thing does not work at all. We're not going to get this happening. So this is why, oh, I forgot to make a 1 over here, didn't I? This is why our homologous chromosomes have to line up so that we make sure we get the right complement of DNA into each of the resulting cells. So what is going to result out of this? I'm just going to draw it for this one here, and I'm going to ask you guys to also draw it for this one. So to draw what 2 cells come out of this. So let me draw it for this one here, and we're going to get 2 cells out. Okay, this is our first round of cell division in meiosis, also known as meiosis 1. So we're going to get chromosome 1, chromosome 3 from mom, chromosome 2 from mom goes over into this cell. Chromosome 2 from dad, chromosome 1 from dad, and chromosome 3 from dad. Okay, so this is going to be our starting point for the next round of divisions in meiosis. So this is what happens in meiosis 1. So meiosis 1 is this set here. We get our homologous chromosomes lining up and then a separation of those chromosomes. Keep in mind, at the end of meiosis 1, every cell is going to still have 2 chromosome 1 sisters, 2 chromosome 2 sisters, and 2 chromosome 3 sisters. So after meiosis 1, this is what we have right here. We've got 2 cells, and each of these cells has a full set of chromosomes, right? 1, 2, 3, 1, 2, 3. The number of chromatids they have is going to be really similar to what we start out with, right? They've got 2 1 chromatids, 2 1 chromatids, 2 2 chromatids, 2 2 chromatids, 2 3 chromatids, 2 3 chromatids. The big difference here is that each of these sets of chromosomes, so 1, 2, and 3, each of them is only going to be either from mom or from dad. Mom, mom, dad. After meiosis 1, what happens is very similar to what happens in mitosis, which is that we just get separation of these sister chromatids. So let's draw the 2 cells that are going to result from this division. We're going to have a division plane that's going to separate out these 2 sisters from each other. So the sisters stuck together all through meiosis 2, but now the sisters are going to be separated to make 2 daughter cells. So daughter cell number 1 is going to get this half of the genetic material. Daughter cell number 2 is going to get this half of the genetic material. And you're going to see that these daughter cells, this is 1 and 3, 1 and 3, these daughter cells are going to look very similar to each other. They look similar to each other. They're identical to each other because we left out one important thing, which is recombination, that I'm not going to talk about in this video. This one, we're just following those chromosomes. So our sisters separate. So each of these two cells that are the products of meiosis 1 are going to divide into two more cells. Woo, that's a weird circle. And so this one here is going to have 2 from the mom. And then 1 and 3 is going to be dad's genetic material. Again, this is with no recombination. With recombination, these chromosomes are going to look really exciting, but we're not going to cover that today. So these are the products of meiosis 2. So that's what we're looking at here. Meiosis 2, we separate the sisters. Meiosis 1, we separate the homologs. Meiosis 2, we separate the sisters. That's it. Hopefully you can get through this video, draw it out yourself. It'll be easy for you to remember.